Hi, everyone. My name is Sherry Xie. I'm currently a bioinformatician in the Mayan lab at the Icahn School of Medicine at Mount Sinai. And today I'll be introducing and demoing 3 Appyters that our lab has developed. So just for some brief context, there are currently a large number of publicly available transcriptomics data sets that have high potential for reanalysis, and our lab sought to repurpose volcano plot visualizations to study how the expression of a single gene is actually modulated by different perturbations across these existing gene expression studies. So to that end, we developed 3 Appyters for querying single genes against thousands of perturbational and disease signatures derived from reprocessed transcriptomics datasets. And these are the MyGenes transcription and type 2 diabetes transcriptomic signatures Appyter, the GeneCentric Geo Reverse Search Appyter, and the RNA Seq-like GeneCentric Signature Reverse Search Appyter. So the first Appyter is the type 2 diabetes Appyter, or the T2D Appyter, which I will call it for short. And this allows users to query a single gene against a database of diabetes and metabolism-related gene expression signatures. Over 300 human and mouse signatures from a total of 119 geo studies were manually extracted by Dr. Kathleen Jagodnik, formerly of our lab, in collaboration with Dr. Mark Keller and Dr. Alan Addy of the Addy Lab at the University of Wisconsin. These samples were then processed using the ArcGIS-4 pipeline, and the signatures were computed using the lemma-R package. Now, I'll go ahead and demo that for you. So starting from the Appyter catalog, if you go into the search bar, you can go ahead and just type in T2D. You'll see it here, click on it. You can start the Appyter. And then today, just to go along with the theme, we will look at the clotho gene in human signatures. Go ahead, submit. And you'll see the first volcano plot, which just shows the expression of clotho in each of the signatures in the database. And the position of each point corresponds to its fold change, which is the X position, as well as its p-value, which is the Y position. And each of these points is a separate signature. It's a bit difficult to tell, but there are square points, which represent microarrays. And then the circular points represent RNA-seq signatures. There's also a table that displays these results. And then in the table, there's some more numerical information, as well as a link to the original geo study, which if you click, it'll just take you to where these samples were derived from. As well as if you click on the link to the bulk RNA-seq analysis, this actually links to this is in the way. This actually links to a pre-processed bulk RNA-seq analysis of the actual samples used to compute that particular signature. And this is also a different Appyter built by Dr. Minji Jun in our lab that provides some visualizations, clustergrams, and differential gene expression analysis, along with other analyses of the actual samples used. Another feature of this Appyter is that if there is a homologous gene in the alternate species, so in this case, we select the human species, the Appyter will automatically identify if there's a homologous gene in the mouse species, and then it'll actually perform the same analysis for the mouse signatures as well. So here you can see all the results for clotho in the mouse signatures. Returning here. The next Appyter is the GeneCentric Geo Reverse Search Appyter. And this Appyter provides a database of automatically extracted signatures from geo studies that were extracted from ArcGIS-4, which is presented earlier by Dr. Alex Lockman. And this Appyter is actually built by Kerwin Shin, who was a 2021 undergraduate summer research fellow in our lab. So in this case, the bulk RNA-seq samples were automatically extracted from ArcGIS-4 and identified based on their single cell probability. And then labeled as cases or controls automatically based on the frequency of the terms found in the actual sample labels. And this resulted in a total of 8,000 signatures that were computed using lemma. And in this case, I'll go ahead and demo this Appyter. So once again, we start the Appyter. We have, again, the choice of species for continuity's sake. I'll go again with clotho in human signatures. And then we can see it is a very similar volcano plot where you have the position of each signature determined based on the fold change in the p-value corresponding to Clotho in that signature. And then once again, you have the tables. These tables also feature a score, which is the confidence of the signature based on the automatic steps that were taken to compute the signature. And the lower the score, the more confident we are in that the signature is valid and accurate. And then finally, we have the RNA-seq-like signature reverse search Appyter, which allows single gene queries against RNA-seq-like perturbational signatures derived from the Lynx L1000 datasets. So these signatures were actually transformed from L1000 expression vectors to RNA-seq-like expression vectors using a deep learning pipeline developed by Dr. Minji Jun in our lab. A cyclogram model was first used to transform the 978 landmark L1000 genes to RNA-seq-like expression vectors. Then a fully connected neural network, artificial neural network, is used to extrapolate the expression of 20,000 total genes. And just for efficiency's sake, because there are 1 million total signatures in the L1000 database or dataset, only the top 100 CRISPR knockout and chemical perturbation signatures for each single gene are actually returned in the Appyter. And then signatures were computed with the characteristic direction instead of lemma in this case. Once again, we have a similar input form for the gene of interest. Go ahead and enter Clotho. You can also select the cell line, but in this case, because we want to see signatures from all cell lines, we'll go ahead and leave that blank. And then once you submit it, you'll see that Clotho is an inferred gene, so it is not one of the original 978 landmark genes. You can click on this to see Clotho and the gene and drug landing page aggregator, which Max showed earlier. And then we can scroll down and see the volcano plots. So here, each point represents a single signature. And the X position corresponds to the full change of Clotho in that signature, while the Y position corresponds to the absolute value of the CD coefficient of Clotho in the signature. Then once again, we have tables featuring all of the metadata associated with the signature from the original L1000 signature. And then we can also actually run an enricher analysis on the top 20 CRISPR targets from the CRISPR knockouts, the top 20 CRISPR knockout signatures where Clotho is up or down regulated. See the list here. And then similarly for the chemical perturbation signatures, because these are all small molecule perturbations, I can actually also run a drug enricher analysis of the top 20 compounds that up or down regulate Clotho in these signatures. And with all of that, I'd just like to thank all the members of the Maya Lab, and I will be available for questions in the breakout room. Thank you.